Hello, everyone, and welcome to today's webinar, Cutting-Edge Viral and Non-Viral Delivery Platforms for T-Cell Engineering and Beyond, presented by Dr. Namritha Ravinder, Senior Research and Development Manager at Thermo Fisher Scientific. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. This presentation is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please click on the Thermo Fisher logo on the left side of your screen. The presentation you are about to see was previously recorded, but I would like to remind you this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speaker will be answering your questions via the email address you provided at the time of registration. Now, if you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or you may use the Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce today's speaker, Dr. Namritha Ravinder. Dr. Ravinder is a Senior Research and Development Manager at Thermo Fisher Scientific in Carlsbad, California. Dr. Ravinder and her team are focused on developing platforms and workflows for cell and gene therapy applications, including gene delivery and viral vector systems. Prior to her current role, she led product development initiatives around mammalian genome editing and functional genomics screening, as well as helped establish a variety of custom service offerings within Thermo Fisher Scientific, including cDNA library generation, virus production, gene expression profiling, and custom RNAi screening tools. She earned her doctorate in biotechnology from the University of Alabama in Huntsville, followed by a postdoctoral research at Children's Hospital in Los Angeles in HIV Virology. Dr. Ravinder will now begin her presentation.
Good afternoon, everyone. Can you all hear me? It's a relatively small group, so we should be, let me know back there if the voice is clear. Okay, good. So thank you all for joining us. So today I'm here from San Diego, California, from the Carlsbad site, here to give you a talk on cutting-edge gene editing and delivery technologies that we have at Thermo Fisher in our portfolio in cell biology. So I'm a R&D leader, currently leading the delivery platform team in Carlsbad. And before then, I was part of the genome editing portfolio group, leading genome editing tool development. So today's agenda, we'll be covering gene editing tools, just briefly going over what we have, and then primarily focusing on CRISPR-Cas9 editing tool technology, and then focusing largely on what it takes to optimize the workflow and the kind of delivery reagents that we have for different payloads and cell types. And then I'll end the talk focusing on some of the viral production systems that we have for research use as well as clinical research use for producing lentivirus and AAV virus. And then we'll close it out with some closing comments on the summaries. So the team that is developing gene editing tools and the delivery reagents is located in Carlsbad, California. And that is a hub for a lot of different portfolios within Thermo Fisher, including the sequencing technologies, reagents for stem cell biology, sequencing, antibodies from the legacy Affymetrix side, and a lot of custom R&D services for cell engineering and cell therapy research workflows. So before we get into the talk, because it's focusing largely on gene editing workflows, I just want to go through briefly on the workflow for gene editing. As most of you might know, and for those who don't, I just want to briefly touch on this workflow. The way the gene editing tools work is you first design a target sequence specific gene editing moiety that would, once delivered into the cell, would find the complementary sequence that it is supposed to bind and cut. And once it binds the sequence that we are intended to change, the enzyme would bind and make a double-stranded cleavage or single-strand, depending on the kind of tool that you're using. And once the double-stranded cleavage is made at a specific sequence, the rest, you're leaving it to the cell's repair mechanism, where it can leverage two major repair mechanisms, one of them called the non-homologous end joining mechanism, where the double-stranded break region can come back together, and during this process of repair, it's largely error-prone, creating a lot of frame shifts, thereby knocking out the gene's function. The other repair mechanism, called as the homology-driven repair mechanism, that can be leveraged in the presence of a donor DNA, where you would insert the sequence that you want to use for repairing the mutation or what have you within the cells. So once in the presence of that DNA, the homology-driven repair mechanism kicks in. So what it does is, now you have a double-stranded DNA, you have a donor, a repair donor, and that donor can be stitched into that double-stranded region, making precise edits that you would like, either a SNP change, you could add a promoter of your choice, or an exogenous DNA that you want to express inside the cells of choice. So those are the major repair mechanisms. There are other types. And then at Thermo Fisher, the gene editing tools that we provide, we have two major ones, TAL and nucleases, and the CRISPR-Cas9 nucleases. I'll be focusing mostly on CRISPR-Cas9, and I'll go through the biology for some of you who may not be aware of this. And then we also provide gene synthesis services through GeneArt, where you could provide your sequence of interest that you want to use for correction, for knocking in a particular gene of interest or a mutation, and then that particular DNA can be synthesized, either as fragments or as plasmids. So now within this workflow of gene editing, if you divide those workflows, you have three major categories. You're designing the payload, right? Either the Cas9 or the TALENs, what have you, or the donor DNA. You design it, and then you order that particular reagent, and then you're delivering it. The piece of delivery is very critical. You really need to make the right choice of delivery reagent. You're either looking at your cell types, or you're looking at the payload that you're trying to deliver, either the DNA or a protein version or RNA that you're trying to transfect into the cell. And once you have used an efficient delivery reagent, then the next step is to wait for those payloads to do its job, and then you're detecting and then using it for downstream workflow. So now if you look into this workflow in more detail for gene editing, after using the editing tool, you're preparing them. The next step after transfecting is you're looking at the editing efficiency inside the cell. And then, depending on your workflow, you're using the cells that have been edited for a disease model, or you have a drug discovery path that you're trying to take using a library screening. Either ways, your main goal is to maximize editing efficiency. And why would you want to maximize editing efficiency? Let's take the scenario of making disease models. If you have a pool of cells, higher the editing efficiency, higher the success rate of finding the edited clone that you want, so the less number of clones to screen. So that is the key. And to do this, it's very important that you have the right payload that will enable you to have the highest editing efficiency, the right protocol, the right media that is suitable for the cells, and the right delivery reagents. So what we do as we are developing these reagents, one of the key focus for the R&D team is to make sure not to just develop the tools, but also ensuring that we have cell type specific protocols with all of the medias and the transfection reagents and downstream analysis. So that is the goal. And that's where we spend most of our time in doing our product development, to ensure that we get really robust protocols, not just the product that goes into the customer's hand, but a protocol that is suitable for our customers. So you will see some examples for what we have done, like case studies of where we have used these kind of approaches for having optimized editing efficiency for stem cells or T cells and so on and so forth. So before we get into that, since we will be touching about different aspects and factors within the Cas9 system, I want to briefly go through what the CRISPR-Cas9 system is. This is a bacterial adaptive immune response-based gene editing tool where the scientists, to keep it brief, the scientists have leveraged this system that drives immune response in bacteria against viruses. They've taken that and used the components from there for mammalian genome editing and several different host genome editing, including plants and other hosts. So the two main components that are needed for gene editing in CRISPR-Cas9 is that purple bubble that you see, the Cas9 enzyme, and the yellow hairpin loop structure that you see there, the guide RNA. The Cas9 is the enzyme that actually helps in cleaving the DNA, the genomic DNA of interest. What drives the specificity is the guide RNA. So if you focus on the guide RNA region, you should see two components there. One, the target specific region, the yellow line there, which is about 20 bases specific to the genomic sequence of interest. So if you have a particular gene that you're trying to edit, you find a unique 20 base pair region and then design your guide RNA for that region. The rest of the guide RNA, which is called the tracer RNA, is constant component that is required for functionality and structure. Okay, so once you've identified the 20 bases, it's almost like designing a PCR primer. You deliver that and you deliver the Cas9 either as a plasmid or a protein or an mRNA that can be expressed thereafter. And then inside the cell, the Cas9 and the guide RNA combine together to form what's called as the Ribonucleoprotein Complex. Now this complex is scanning through the genome and it's looking for NGG sequence in the genome. That's called the PAM site, and this PAM site is unique. The NGG sequence is unique for the CRISPR-Cas9 system from Streptococcus pyogenes. For other CRISPR-Cas9, the PAM sites are different. So now the Cas9 and guide RNA complex is scanning the genome, sees the NGG sequence, then it's trying to match the upstream 20 bases to the guide RNA to see if the guide RNA is complementary. If it doesn't, it's going through and scanning until it finds the respective complementary sequence within the genome that you're trying to design the guide RNA for. Once it finds it, it binds to that region, the enzyme gets active and makes a double-stranded cleavage with the wild-type Cas9 because it has two cutting sites within it. So now what's really critical, the first step in designing the genome editing tool is designing the right guide RNA. So what we have done, and you'll see very similar, the others in the field have worked a lot on designing guide RNAs. There are different rules for guide RNA designing. The first step is to find, you take every gene of interest within the human genome. If you want to create a library of guide RNAs for human genome, you would look at all the genes, take all possible guide RNAs that are unique sequences, meaning you have that 20 base pairs only at that intended site, but not elsewhere. Otherwise, you will have off target, right? Once you find all possible guide RNAs, you take those guide RNAs that have 5' region if you want to knock out a gene. If you're just silencing the gene, you're trying to focus mostly towards the 5' so that you are cutting that genome early on so that you can knock down the message downstreams. And then once you take those 5' regions, you're also looking at off-target effects. You're trying to look at 1 or 2 or 3 base pair differences, and then will this sequence bind elsewhere in the genome? And you do the off-target analysis, and you're also looking at binding efficiencies of these, the GC content, because sometimes if you have stronger GC content, the binding of the guide RNA to the genome is much stronger. So even if you have 1 base pair difference within that 20 bases and you have a sequence similar elsewhere in the genome, the chances are it'll bind stronger. So there's a fine balance of the GC content. So once you do all of this, what we do for the knockout libraries is we would take the top 4 unique guide RNAs and use that for gene knockout. So you can take 1, 2, or 3 guide RNAs to knock out the gene. So that is for gene knocking out. So we have a library, so one could buy those guide RNAs from the library for a specific gene of interest. But if you're doing knocking in, where you're doing precise cutting, what you need to do is, you're not going to look at the 5' region, you're looking at the region that you want to make the mutation at. Right? And then you will pick that sequence, and then you're trying to blast that sequence and find a unique region. Now we have a tool within Thermo Fisher that can help you to design unique guide RNAs for knocking in. So once you've done these designs, your next step is to pick the right Cas9 format. So if you look at in the field and within Thermo Fisher, we have gone through a journey of developing Cas9s in different formats. Having Cas9 being expressed from a plasmid, So once you have that plasmid transfected into the cell, they'll express within the cell the mRNA and then the protein. And then the field moved to going from the plasmids to the mRNA, because plasmid DNA, there is a concern of random integration. Because if the DNA stays in the cell for too long, it's constantly expressing the Cas9 until the DNA is cleared from the system. Therefore, what happens is the Cas9 does its job, cuts the genome, but it's also there to go and look for other sites. So the chances for off-target are much higher when you have plasmid system. So therefore, the field, and so did we, move to the Cas9 mRNA system, and then to what we have, the industry standard right now is the Cas9 RNP system, where you have the Cas9 as purified protein, recombinant protein, and the guide RNA as RNA. So now you have a DNA-free system that sets you up for success to have highest editing efficiency and also lowest off-target. So now, depending on the kind of payload you use, the delivery reagents and the delivery method will be different. So if you're using a plasmid-based system, you would go for lipofectamine 3000. If you're using mRNA, you would go for a delivery reagent that is specific for mRNA, lipofectamine messenger max, and for protein, the CRISPR-Cas9 RNP system, you would go for a CRISPR-Max delivery system. Those are the lipid-based reagents for transfection. They're really nice when you're doing high throughput screenings and etc., and for relatively less challenging cell types. When you are dealing with primary cells and more difficult and challenging cell types, then you would want to use the electroporation system, and what we have is a neon electroporation system, and you'll see some examples of using this in primary T cells and iPSCs and so on and so forth. We also have viral-based delivery system, and you'll see those examples later on. We have lentiviral system, as well as in development, we also have an AAV, Adeno-Associated Viral System, production systems to make viruses that can be used as gene delivery vectors for different cell types of interest. Again, the key here is always remember the payload that you're using, and based on the payload, you want to choose the transfection reagents, because if you think of these transfection reagents, the way they are designed is, or developed, is screening those for a fit for purpose for that particular format that you're trying to deliver within the cell. So what we have done, this is just an example to show in our journey of designing guide RNAs, our first goal was to make sure we have highly efficient guide RNAs in our CRISPR libraries. So in that case, when you're trying to design and screen the guide RNA that is having highest efficiency, if you have a Cas9 expressing cell, so you have a cell that already has a Cas9 expressing as a stable cell line, then all you're doing is you're delivering a guide RNA into the cell. So you're just delivering the RNA. So you use a lipofectamine RNAi max reagent that is specifically designed to deliver RNA into the small RNAs. In this case, the guide RNAs, which are about 100 nucleotides in length. So this is an example just to show that we have tried in this particular cell line different guide RNAs to see what is the editing efficiency for that particular guide RNA. Now, the next example here is just to show that for, as I mentioned, for plasmid DNA, it's lipofectamine 3000. This is just showing you that we at Thermo Fisher, we did have different older versions of transfection reagents like lipofectamine 2000. And compared to lipofectamine 2000, when we had CRISPR reagents, we wanted to make sure we developed reagents that are even more robust, and we screened different reagents, and we found lipofectamine 3000 to be much more robust than 2000. And this example is just showing GFP plasmid using one of the competitor reagents in the field, Fugene, alongside lipofectamine 2000, our older generation plasmid DNA, and then the lipofectamine 3000. Again, as I said earlier, plasmid DNA, we have very good transfection reagents, but when you're doing gene editing, you really want high gene editing efficiencies. So it's always best to go and use the Cas9 ribonucleoprotein RNP system, where you can deliver a Cas9 protein as a purified recombinant protein, and the guide RNA as RNA. And the reason why we would like that is, if you think about DNA, the DNA has to enter into the cell, the Cas9 has to express, and then it has to find the RNA and in the right 1-to-1 molar ratio. So you're leaving that ratio, finding the right ratio to the cell to decide. Same thing in mRNA. When you deliver Cas9 mRNA, it gets into the cell, it has to express the protein, and the protein has to find the guide RNAs in 1-to-1 molar ratio. In case of the Cas9 protein and guide RNA, when you're trying to transfect it into the cell, you already have the protein ready to go and act. And before delivering it into the cell, you can complex the Cas9 protein and the guide RNA in 1-to-1 molar ratio and form the RNP complex in your reaction tubes. So you can control that molar ratios. You're setting yourself up for success before transferring that into the cell. So it is ready to act, and you have it in the right molar ratio. And in addition, it doesn't have the same threat like the plasmids of randomly integrating into the cell, right? And also what you could do with the protein, you can just give enough amount of protein needed for optimal gene editing. And what you will see on the right side, the Western blot there, shows you, this is an example from 293, just to show you the amount of protein that is expressed with the DNA. So you're normalizing the DNA, mRNA, and the Cas9 protein to the equal amount of editing efficiency. So you have the same amount of proteins when you're delivering. And if you have your DNA, what's happening is the DNA is constantly expressing the protein. From 0 to 72 hours, the level of protein is increasing in the cell. So what that means is, after the editing happens, what we notice is as early as 12 hours to 24 hours, sometimes depending on the cell type. So the editing happens and the protein is still left over to go and attack other off-target sites. So you're increasing the chances of off-target more with the DNA version. And if you look at the mRNA, it remains constant because you're controlling the dose of the mRNA. Protein, you deliver the protein, you have a high peak of the protein in the beginning because you're adding that into the cell, and as time passes, it turns over. So what that means is it does its job, and the protein is then removed from the cell much faster. So you would want to go with the RNP system to increase your efficiencies and then clear the protein from the system so that you don't allow it to do any off targets. And down below, the graph there actually shows examples of 2 different off-target sites for the specific guide RNA we were using in this case. So what you see here is with the DNA, you can see much higher editing efficiency of both the off-target sites, whereas the protein barely has any cutting efficiency at that site. So in this experiment, what we're trying to do is you're intentionally designing a guide RNA that's not that unique, but has potential off-target. You're designing a bad guide RNA to just look at the off-target effect. In reality, when you're doing your experiment, you wouldn't do that, right? This is just to show that the DNA-based approach has higher off-target activity than the protein. So long story short, what you really want is design a good guide RNA and go with the Cas9 RNP system to increase your editing efficiency. Here are just a whole bunch of examples to show that the Cas9 RNP system, when used with the lipofectamine CRISPR-Max, again, this is the lipid-based reagent, you can cover most of the cell types. Examples shown here are A549, the adherent cells, some suspension cells like THP1, and then a couple other adherent cells at the bottom. So pretty robust editing efficiency across different guide RNAs or different gene sequences that we have tested. This is one example showing HPRT, RELA, and AAVS1, 3 different genes that we were trying to target. HPRT and AAVS1 are intronic regions, and the RELA is a exonic sequence. And the first graph, what I'm trying to show you here is the comparison between the plasmid-based system and the Cas9 RNP system, where you're using the Cas9 protein and the guide RNA. And you can see HPRT is a really robust site, and with the DNA, when you get into really tough cell types like HG2, the suspension cells, with a really robust delivery system, which is neon electroporation, you still cannot get over 10% editing efficiency in these cases. Whereas with Cas9 protein system, you can almost go up to 90% editing efficiency, depending on the target type. Just to show you an example that more difficult cell types, you start using the plasmid, the efficiency is not that great. You want to switch to the RNP, not only for off-target, but also for highest editing efficiency. On the right side, the graph is just showing an example of lipofectamine CRISPR-Max with Cas9 RNP. And we have optimized the media, the protocols, and we have a fit-for-purpose protocol for this particular case. And you're comparing with equally good competitor reagent and trying to see how we perform. And what we see is when we have optimized protocols, our RNP system with our robust protocols that we have works much more efficient than what's out there in the field. Now, what I want to show next is the real advantage and the use of these editing tools is in CAR-T therapies or making isogenic disease models with iPSC. So for those of you who are not aware of what isogenic disease models are, in case of iPSCs, you can take a wild type cell, you can differentiate that into whatever tissue that you're interested in studying the disease model. And then you can take the same wild type iPSC cell, create a disease mutant, and create an iPSC that is a mutant variety now, and then now differentiate that into the disease model with that specific disease relevant mutation. So now you have, from the same parental cell line, you have the wild type as well as a mutant disease model. So you're creating an isogenic disease model from the same genomic background, as opposed to taking two different patients who are different, coming from different genetic backgrounds, different regions. You have much more complexity there. So the beauty of gene editing is really that you can create isogenic models using iPSCs and then do your comparative studies in that background. So I just want to show you an example of what we have done during the development process. So we had our Cas9 reagents, we have different medias and assay systems specific for iPSCs and different transfection reagents that are specific. So we have lipofectamine stem, lipid-based reagent for Cas9 RNP transfection, and then of course neon to boost the editing efficiency even more. So what we have done, our first journey in developing the protocol for iPSCs was we used the neon. After optimizing our conditions with lipid-based system, we moved on to the electroporation system, the neon system. The beauty of the neon system is it has 24 predefined electroporation conditions with different voltages and so on and so forth. So when you are beginning to optimize your condition, you can take a control guide RNA. We do offer different controls like HPRT and others. You can take a control guide RNA, use the right media system in the transfection conditions, test all of the 24, pick the best transfection condition. In this case, we have picked a few different conditions, and then what you see here is the optimal condition for DNA, optimal condition for mRNA, and different dosages of protein with a specific condition that we have optimized for electroporation. And as you can see, the editing efficiency with Cas9 RNP system is much higher compared to the mRNA or DNA, even after using the best conditions for each of those. So this is just to give you an example that you really need to spend a lot of time upfront to optimize your conditions, even if you have the best editing reagent or transfection reagents. You want to make it cell type specific. So that's where we spend most of our time. Here is another example showing two different graphs that you'll notice here. One in red is the indel that is knockout. So you're using Cas9 RNP system with electroporation to knock out the gene, cutting a specific gene. So there are different types of genes here. And the second, the purple graph that you see, is for that same site, you're inserting specific mutations. So you're doing precise gene edits at that locus. So first, if you look at the knocking out efficiency, depending on the target that you are, it's also target specific, the editing efficiency. So it's not just the reagents that you use, but also the target sequence that you're trying to edit. And that's one of the reasons why we always recommend our customers to design at least 2, if not 3 guide RNAs for each specific locus that you're testing and pick the one that is the best. Okay? And similarly, when you go for, even though you have really high gene knockout efficiency, you still have to make sure that your knocking in efficiencies are good. And if you look at the HDR efficiency, again, depending on the site and the sequence that you're trying to edit, the editing efficiencies can vary. Anywhere between 20% to 80% for knocking out, and then 5 to 40% for knocking in. So what we have been doing now that we have all these good reagents and workflows, our focus is to really boost that knocking in efficiency, because that is where the field is moving to make precise edits. We are trying to identify new molecules that can boost editing efficiency by electroporation, and that's where most of our work right now is focused on. So the next example that I want to focus on here is T-cell, primary T-cell editing. So before the RNP times, we barely were getting any good editing efficiency. It was really single-digit numbers. So with a good RNP system that we have and a good condition of neon electroporation, what we then decided to do is build a workflow-specific protocol. So in this example, we used our CTS grade, the cell therapy systems grade Dynabeads to isolate the T cells. And we also have T cell activation, bead-based activation kits for isolating T cells as well as activating T cells. So you really need to activate the T cells before you edit. So after 3 days of activation of the T cells that have been isolated, you then transfect or electroporate the cells, the primary T cells, activated T cells, with the Cas9 RNP. Okay, so in this case, what we're trying to do is, if you're familiar with the CAR T space, the TCR receptors are one of the favorite target sites for editing. So here we have used 2 different guide RNAs for TCR alpha chain and 2 different guide RNAs for TCR beta chain. As you can see, depending on the sequence of that site that you're doing, the editing efficiency can vary. So once you've selected the best guide RNA, then you go back and do your real experiment. So the first, what we do is we want to make sure the T cells that we have isolated are really T cells. So I'm just showing a snippet of the data here. So in this case, we are looking at, if you have really good T cell isolation, you should have greater than 90% TCR positive cells. I'm doing flow cytometry here using antibodies against the TCR receptor. So in an untransfected cell, I should have over 90% TCR positive, and that's what I see in my untransfected sample. Now, when I use a Cas9 RNP that is specific to TCR receptor, a guide RNA specific to that receptor, and if I have high gene editing efficiency, I should see a leftward shift of all of those cells. So TCR negative population. So as you can see on the rightmost graph, we really have over 90% editing efficiency, which is really strong efficiency right there. And that's what you want, because ultimately you're trying to add these engineered T cells into a host cell, and you want to maximize your chance of getting the effect that you intend to see. So this is just knocking out a gene. The beauty of gene editing is, again, by making precise edits. So then what we did, we also wanted, before we did specific mutations, we also wanted to see the efficiency of multiplexing. So in this case, we are trying to knock out TCR gene, B2M gene, which is specific to the MHC complex, which is also another favorite target that is knocked out for CAR T workflows, and then PD1. So these 3 targets, what we're trying to see in this experiment is, if I transfect these guide RNAs individually versus I divide the total amount of guide RNA that I'm transfecting, divide that into 3 different guide RNAs, will I get equal efficiencies, or do I need to boost the amount of guide RNA for each? So if you look at the graphs here, what you will see, the purple graphs right there are single guide RNAs. That means each well, I have only 1 guide RNA for that particular cell type. And then the red bars, I have another well with all of the 3 guide RNAs pooled together. So basically, you're taking the Cas9 and all 3 guide RNAs into 1 tube, mixing them. You're dividing the total amount of guide RNA molar amounts that you have into all 3, and that's the 4th well. And when I'm doing my editing efficiency post-transfection, 48 hours later, I harvest the cells, and I'm doing NGS sequencing, either taking that single guide RNA and sequencing that specific locus for that single RNA, or taking the pooled samples where I have all 3 guide RNA, and then doing the sequencing for all 3 targets. And as you can see here, whether I transfect as single guides or I have only 1/3 the amount in multiplex, I have equal amount of editing efficiency. Again, long story short here, the message that I'm trying to deliver here is because of the efficiency of RNPs, you really don't need that much amount of RNP. Now you can multiplex your experiments. We have tried up to 6 guide RNAs in a single experiment and then looked at editing efficiency in multiplexing. So if you see again the flow cytometry experiment, I start off with strong TCR positive and B2M positive cells. As I do my knockouts, I should start seeing TCR negative and B2M negative population right there. And I'm getting close to 70% multiplex samples with all 3. You don't see the PD1 here, but we're getting over 60% with all 3 guide RNAs knocking out all 3 sites. So the next one now is looking at the knocking in efficiency. So in this case, we are taking T cells and trying to add a orange fluorescent protein downstream of an actin promoter, okay? So we are trying to knock out the beta-actin site and then cut that site and add a 1.4 kb OFP gene sequence. So if you notice this, controls again, this is at the TCR site, I'm sorry. So in this site, you see TCR positive to begin with. And now I'm trying to knock out the cells. I should see TCR negative population, which is pretty good by knocking out. Now in the knock-in sample, what I see with the regular transfection electroporation buffer R, I'm barely getting any knocking in efficiency of my OFP protein, which is about 5%. So what we have done then is we wanted to improve this efficiency. We screened several small molecules that can boost the knock-in efficiency. And what you see in the rightmost graph, flow cytometry data, is by using this delivery buffer called R buffer with a small molecule in there, that is boosting the editing efficiency from 5% to close to 20% knocking in efficiency. So we are able to increase by several folds the knock-in efficiency. So these are the kind of efforts that we are doing within R&D to see what else can we do to boost knock-in. So using cell type specific delivery reagent, Cas9 RNP, and the R buffer with small molecule that can boost knocking in efficiency, we are able to get over 20% knock-in efficiencies. We are doing similar type of experiments for different cell types to see if we can have cell type specific buffers with the right formulation of the molecules small molecules to boost editing efficiency in different cell types. So again, several different papers that you will see for neon conditions, but I'll go into the last 2 to 3 slides that I have is changing gears from Cas9 RNP to the viral vector production system. Again, the gene therapy world, the lentivirus and the AAV systems are pretty favorite in the field. And the goal there now for us is to provide production systems with the cells that have been screened for lentiviral production or AAV production, with the right transfection reagent, right media, and the complexation buffers, so that our customers, when they buy that combination of kits, you can have a good production system to boost your titers. So we launched what we call the LV Max system as a research grade use product, and then followed that with the CTS grade for cell therapy systems for your GMP workflows. So customers can use this particular system and scale up to make large-scale lentiviral production. So you can do anywhere from 96 well format to 2 liters or bioreactors larger than that to produce virus titers of 1 × 10⁸ or more. Okay, so again, from our experiences that we have learned from the lentiviral system, we now then want to go into the Adeno-Associated viral systems, and what we are working on right now that's in development is the AAV Max system. So what we have done here, these are all suspension cell-based production systems. So we have taken our XP293 cells, screened them, and picked clones that are effective producers of AAV, and then used those cell types. So you have in this kit suspension cells that have been built specifically for increasing AAV production. And then we have enhancers to boost the transfection so that you get good titers towards the end. We have a suitable transfection reagent that have been specifically formulated, and the protocol is optimized for having really robust titers of AAV. And then along with this, what we have also done is some work on the lysis buffer. So usually in the field where people are making these AAV, what they do is sometimes they have to centrifuge the cells and then isolate the harvest. Think about bioreactors that are large. You cannot do the centrifugation. So what we have done with the lysis buffer is directly adding this lysis buffer into the culture. So you can lyse the cells in cell culture and then use it downstream for either TFF or other downstream process. We are also working with our collaborators within the company within the purification side and trying to build that protocol from all the way from producing the virus to getting the right protocols for purification using our porous columns. So not only building the production system for virus production, but also having the workflow from end to end. So again, this is a scalable suspension system, animal origin free. Right now we are in development, but you can get early access to this production system. We are offering the beta version for beta testing if you want to do your PD work, process development. And then we will support you through the journey from all the way to the CTS version for GMP workflows when we have that. So I'm going to skip this slide and go to the last one. This is just to show you an example. Again, AAV, there are several different serotypes depending on the tissue that you want to target. So what we have done is after optimizing and building this production system, just like you have seen, we spend a lot of R&D and research to really make fit-for-purpose protocols. And in this case, you're seeing using our in-development product that we have, we've tested it across different serotypes, AAV2, AAV6, DJ8 and 9. And what you're seeing in this graph is what people have been doing is mostly they use HEC293T systems. And we are comparing that to the suspension version with the AAV Max. And as you can see, with 1.5 microgram per well, in this well format, you can get equal or better than the HEC293T systems. So here, the message here is, as we work in product development, it's not just to get that reagent, whether it's a transfection reagent or viral production system, in the hands of the customer. We do understand the pain points from designing an editing tool, if it is gene editing with CRISPR-Cas9, all the way to different factors like media, transfection reagents, and downstream processes. Very similar for the viral production. We are going through that journey of making sure we have the right protocols to get the really high titers, but also making sure that the downstream purification protocols are fit for purpose. So that's where we are focusing our efforts. So if you have any questions, I'm still here for another, we have 15 minutes more for Q&As, and I'm happy to answer and do some deep dives. If you're interested, I'll be outside the theater for any other questions that you have. And with that, I would take some questions that you might have.
A big thank you goes out to Dr. Ravinder for her excellent presentation. Now to our audience, if you do have any questions you would like to ask, please type them in the Ask a Question box located on the far left of your screen. Dr. Ravinder will be answering these questions via the email address you provided at the time of registration. I would also like to thank Labyrinth and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labyrinth will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.